Okay, let's start. Good morning, afternoon, and evening. Hola, hello, bonjour, hola. Ni hao, konnichiwa, 안녕하세요, and hello all. Welcome and thank you, SynBYSS people, for joining us the 145th seminar, and I call it UC Davis Day. I am in Brno, Czech Republic, attending a synthetic biology conference with my student after visiting Germany and Austria to meet my colleagues for potential collaboration. It was great to see many of my colleagues, including, of course, mostly European people here. Okay, it is my tremendous honor to introduce very briefly. I will try. It's probably not easy to briefly introduce our pioneer speaker, Professor Cheemeng Tan. He is a professor at University of California, Davis, and also Chancellor's Fellow. I got to know him for the first time because of his amazing paper with Ling Chong Yu on a burden-associated biostability circuit published in Nature Chemical Biology, which I use for my SynBio course for 10 years. After he started his professor position, he continues to amaze me with numerous great, publications. And that's why I really wanted to meet him. And then finally I met him in conference, I believe, many years ago. And Chimeng, thank you so much for your inspiring work and effort to educate our future leaders, and the virtual podium is all yours now. Thank you again.
Thank you. Yeah, thank you, Taisha, for the introduction. Yeah, my lab nowadays, thanks for the introduction on the paper that I published with Ling Chong. It has been many years since then, and we now work a lot on trying to engineer synthetic life against disease. So just want to introduce my lab briefly. So we primarily work on two directions that kind of align with what has been going on in the field on drug delivery and therapeutics. There are really two major chassis in the field. One will be along the line of particles, liposomes, and another will be cell-based. So in the direction of nanoparticle systems, these are primary material-based systems. They're minimal, but the challenge is they usually lack sustained activity in vivo. And when you deal with living cells, then you're trying to deal with very complex systems, and they often have various issues from mutation, immune response, or stability in vivo. Now, my lab interests really to combine materials and nature component as a new system or synthetic system that could potentially be superior than the classical chassis. So one motivation that one direction that motivates our work is this idea that cells secrete this exosome or extracellular vesicles in nature for cell-cell communications and also modulating immune cells. Now, engineers have always been interested in harnessing these extracellular vesicles for therapeutic applications. But the challenge is these systems are so complex and heterogeneous. How can we actually build this kind of a synthetic extracellular vesicle from the bottom up? So that's where my lab combined a series of various tools that we have developed over the years to kind of enable this workflow, all the way from identifying unique protein features from extracellular vesicles in nature, and then leveraging those features and then build synthetic vesicles that mimic physical and chemical properties of those vesicles from the bottom up, using cell-free protein synthesis and also high-throughput robotics and microfluidic systems. And this has allowed us to to be able to run more than 30,000 reaction rapidly to prototype nanovesicles. And after building this vesicle, we have a range of tools where we look at these 100 nanometer vesicles in details and then test them in vitro and in vivo with different kind of disease models. And this is really thanks to collaboration with quite a few labs in UC Davis with various expertise from extracellular vesicles to organ-on-a-chip devices. In another direction, my lab is interested in solving these issues of sustained therapeutic in vivo. And like I said earlier, when you have nanoparticle systems, they are easy to manufacture, but they lack sustained activity. But when a cell-based system, they can deliver drugs in the it's slightly longer than the nanoparticle, but they often lack stability. And you also have to worry about uncontrolled proliferation. So we need a new paradigm, and this is my argument. This is how I organize the field. So when you have full or fully or autonomously replicating systems, this is what synthetic biologists usually deal with. It can be bacteria, hack cells, all the way to primary cells. You want all these functions: chemotaxis, protein synthesis, dynamic response, and they have all the function you need. But when you go to another extreme, you stop the growth, you also kill the systems, they lost all the activity. And there's a bunch of technology in between, and that can be from attenuation, autotroph, or you use some kind of drug to kill the cells so they can last for a while, but they eventually die very quickly within a few days. And if we can achieve this space where you have non-dividing but active systems, and we could potentially have safe and active system that can serve long-term in vivo as therapeutics. Now, naturally, some cells can reach there, like neurons, some retinal cells. They can differentiate from wild-type cell to the non-dividing state. But really dealing with those cells is not easy and not easily scalable as well. So my lab created a method that we call SynBYSS. Essentially, we create JLO inside the cell. And in the optimal level, you create this kind of semi-living systems, non-dividing but metabolically active. In this previous work, we have shown that they maintain the features of the bacteria, and they maintain metabolism, protein synthesis, and they also become quite stable. So become a kind of a superpower cell that resists high pH, hydrogen peroxide, a bunch of antimicrobial agents. And we're right now extending this to different kind of cell lines, all the way from bacterial pathogens to primary mammalian cells or human cells. And we're currently testing this for various applications that it's very common in biomedical sciences, all the way from cancer to regeneration applications. So thank you. That's a quick intro. So my lab work on artificial nanovesicles, SynBYSS from the synthetic biology, system biology approach. And thanks to the lab members and also the various funding sources. Thank you so much.
Thank you. That's absolutely amazing. I remember your, the work about that Jello thing. That's a fascinating idea because I do care about biocontainment, as you know. This gonna be so I mean, you don't need to worry about biocontainment with this system. So I actually love your work a lot. So
Thank
I'm
you.
happy to see again your talk. Fabulous. All right, so let's go to the next one. Okay, so the main speaker of today with a longer introduction, Professor Sonja Billerbeck holds a master in microbiology from the University of Tübingen, Germany, and a PhD in bioengineering from ETH at ETH Zurich, Switzerland, and after postdoctoral research in East Synthetic Biology at Columbia University, of course in New York City, USA, she joined the University of Groningen, Netherlands, as an assistant professor for molecular microbiology. And she gonna move to UK, Imperial College, in January, and congratulations again. This is another big move. And this basically means she has been all over the world. That's kind of one first introduction part. In Groningen, Sonia develops a research program that uses synthetic biology, protein engineering, and environmental microbiology to access, um, also understand, and also engineer the functional diversity of nature's yeast-based microbiome for applications in human health, industrial biotechnology, and also to answer fundamental questions on yeast pathogenic or just yeast biology. Besides her research, she is dedicated to education and outreach in the field of microbial synthetic biology. She has been leading iGEM teams at various institutes, as I mentioned earlier, for over 10 years, and also co-leads the iGEM engineering committee. Further, she is the chair of microbial biotechnology section of the KNVM, Royal Dutch Association for Microbiology, and also the NBV, Dutch Biotechnology Association. She is also the chief editor, very recent one, chief editor of Oxford University Press Synthetic Biology Journal, as well as an editorial board member of New Biotechnology, where I am the chief editor. I heard, she's very excited to move to the Imperial very soon, but at the same time, very busy at this moment, as you can imagine. So, Sonia, thank you so much for your time today, despite your crazy, probably schedule next, I guess, 2 months. And please take it away. Thank you.
Thank you very much, Tae-suk, for this very kind introduction. And also thank you very much for all the efforts you do and to bring this community together. I think you're very famous for traveling around the world and talking to people. You're very well known for, especially enhancing the voices of young researchers. And I really appreciate that because it's a lot of time and work.
My honor.
While I'm talking, I'm gonna share my screen. Oh, go back. I hope you can all see my screen.
Yes.
So I want to start with introducing my research program that I have been building at the University of Groningen, where I started my assistant professorship and was recently promoted to associate professor. So what we do, or what I call my research program, is like microbial synthetic biology for human health and food security. That's, of course, very broad. And we focus on addressing fungal pathogens. And I want to start by telling you why we do that and what actually the problem is with fungal pathogens. Because when I talk to my students, for example, in my applied microbiology course, what first comes to mind when I talk about fungi is, for example, the occasional mold on a piece of bread you're eating, or maybe, the mold on the wall in your student housing, or some more topical infections like a candidemia in your mouth or a thrush or a foot infection. And what is less known to my students, usually when they start taking my course, is that fungi can really have detrimental implications for humans. So they can cause systemic infections in humans, especially immunocompromised patients. And that causes about 3 million deaths per year, which is more than the death toll of tuberculosis or malaria or breast cancer. In addition, actually, about 80% of all crop diseases we know are caused by fungi. And this in total leads to about 30% crop loss per year due to fungal plant pests. So they also have a huge impact on agriculture. And then even after agriculture, so to say, after post-harvest, about 20% of all food and feed is lost to microbial spoilage. And a lot of these microbes are also fungi. So again, fungi have a huge impact on the food chain, both agriculture and post-harvest. And an additional issue is that we have very few antifungals, so 4 major classes to be precise, and many are only static. And even after treatment, you have high mortality rates because they're not often effective. And in addition, resistance to many of them is already on the rise. So what we really need in this area, next to, of course, stewardship, so we have a similar One Health approach as for antimicrobials, which includes stewardship and basic research, is we actually need new classes of effective antifungals, ideally with new modes of actions and diverse modes of actions. This is difficult because, as you know, fungi are eukaryotes, so it's very difficult to find drug targets that distinguish fungi from us humans, for example, or from plants. Ideally, in addition, we also would need different agents for agriculture, food, and medicine, because what we're currently doing is actually co-using many of these molecules. So, for example, the gold standard azole, the gold standard antifungal in clinical use is also used in agriculture. And this is thought to be one of the major drivers for resistance. So ideally, we have something like targeted biocontrol in agriculture, where we really just target a disease and don't use these like broad spectrum antifungals. And in addition, of course, it would be great to understand the spectrum of resistance mechanisms and have strategies to overcome them. So what we do in my lab now, we have two approaches to directly target fungal pathogens, and that involves a lot of synthetic biology approaches, which is also my background. The first thrust in my lab I call the yeast toxicome. And this is also the thrust I majorly going to talk about today. So these are antifungal proteins which are secreted by environmental yeast. And what we do, we try to access them from the environment, understand how they work, and then engineer them to make them fit for purpose to really then apply them in various sectors. We also have a second research thrust where we focus on fungal G-protein coupled receptors. So G-protein coupled receptors are the major class of transmembrane receptors in humans, but also fungi have GPCRs, and actually fungi have their own classes of GPCRs, and they have not been well understood. And we use the synthetic biology approach to basically understand how they work, but also what ligands are actually recognized and what environmental cues, because it has been shown that many of these GPCRs actually control traits that are related to disease in humans or plants, so kind of virulence traits like biofilm formation, for example. And then we have a third thrust, which actually goes beyond that. So I told you that fungi have a huge impact on the food chain, on traditional agriculture. So what if we could just kind of like invent an alternative food chain? And what we do here, we try to uncouple food production or maybe the production of pharmaceuticals from agriculture by using so-called hydrogen oxidizing bacteria. So this is a recent thrust we started working on, where we have strong collaborations with the company Solar Foods in Finland. And this is all and other research institutes like the University of Aachen and also Ginkgo Bioworks. This is a project funded by the European Innovation Council. And the goal would be to kind of like be able to engineer these hydrogen oxidizing bacteria that can live from carbon dioxide and hydrogen alone, so from air, so to say. They don't need any sugars from agriculture, and have them produce clean food products or in the future pharmaceuticals. Again, I'm going to particularly focus on this first thrust here, but keep watching our space for updates in these other areas. So the yeast toxicome. So what I call always the overlooked talent of yeast. So we know yeast has many talents in terms of, baking bread, making insulin, or making fermented beverages, but there's really this like one talent that is overlooked, I feel, and this is the fact that yeast secrete these antifungal compounds, and these compounds include proteins. And as I by training am a protein engineer, I'm mostly interested in this protein component of it. And when I say yeast, I do not only mean baker's yeast, I really mean all types of yeast which are out there. There's about more than 1,000 species of environmental yeast out there. And again, these compounds can then kill other yeast and filamentous fungi, including those that are pathogens to us humans, but also to plants and those which are food spoilers. And just by numbers, that's a very frequent phenotype. So about 10 to 40%, and I'm going to show some numbers that we produced in our own hands. So 10 to 40% of any given yeast isolate will actually have an antifungal properties. And why we're interested in this so-called yeast toxicome, and this is, I call it toxicome because historically the protein component of these compounds has been called yeast killer toxins. So this is why I now call it the toxicome. It's very interesting to study to me because of different features. So first of all, these toxins that we know, so we know about a few toxins that have been studied about 20 years ago in very detail. So they show various modes of action to kill, so very different types to kill fungi. Ranging from pore formation, so some just punch pores into the fungal cell membrane. Some inhibit cell wall biosynthesis, but some of these toxins really get internalized into the yeast and then can cause cell cycle arrest or have tRNase function. So a lot of different modes of action that I think would be interesting to get inspired by. They also seem interesting from a protein evolutionary point of view, because none of these protein sequences that we have is actually related to each other. So they seem to be evolutionary very unrelated. And we also do not find a lot of functional homologs. Later, I will tell you that maybe we have a foot in the door now to, from a structural point of view, actually be able to classify them more into one kind of evolutionary group. They also seem to be potentially modular. So I'm, as I said, a protein engineer in a synthetic biology. So I'm interested in modularity just because it would allow us to maybe stitch different pieces of these toxins together and make chimeric toxins. So the different functions they seem to have, or different functional domains, is they have often domains that allow them to dock to the fungal cell wall, and domains that then eventually encode the antifungal activity, and also domains that actually protect the host from self-intoxication, because many times these toxins are produced by yeast and kill very closely related other yeast. So the producer needs to have some ability to protect itself. And then I think they're also amenable for engineering, because in the end, it's proteins and yeast, and we have a lot of tools how to engineer them in our synthetic biology toolbox. And if you're interested to read about it, I would recommend our recent perspective in trends in biotechnology, where we explore a bit this toxicome and how synthetic biology could help us to kind of like bring it to the next level of applications. So in a nutshell, now what we're doing, we try to take this natural fungal competition system, bring it into our lab, engineer it, and make it fit for purpose for applications in human health, biocontrol, and food preservation. And how I approached that starting 5 years ago, is really building this pipeline where we access basically these antifungal yeast from the environment using just classical approaches like environmental sampling and high throughput screening. Then I envision to find these toxin encoding open reading frames in the genome of those yeast. That's not trivial, because as I said before, they're evolutionary, not related to each other. So we're not really knowing what we're looking for. And then what we do, we try to understand how the toxic function is actually encoded in these proteins by using high throughput protein science, something like, for example, mutational scanning. And we also try to understand their mode of action. And here we use genome-wide CRISPR-I screens to understand or get a foot in the door how these toxins work and how different their modes of actions are. And then last but not least, I'm interested in engineering them using all the tools that are available to us. And when I talk about engineering explicitly, so we have several engineering goals in mind. So first of all, the natural producers usually produce small amounts of the toxin, amounts that are environmentally relevant to them. But we, of course, in an engineering setting, of course, want to have access to large amounts such that we can, use it in industry, for example. So the engineering goal would here to have just yeast produce a lot of them, a lot of these toxins in heterologous settings. The second engineering goal is actually, so if we look into the natural system, these yeast often secrete one toxin, for example, or they often have multiple confounding traits that in some lead to their biocontrol activity, for example. So that could be, for example, they outcompete nutrients. At the same time, they secrete several different toxins and maybe even a small molecule. So ideally, we can tease all these different functions apart and then have clean engineered strains where we can plug and play these different traits together and make like cocktails, for example, which have defined action. Then we also want to actually go do protein engineering. So these natural toxins often have a very narrow pH and temperature profile where they're active. So often they need very low pH and can only work at certain temperatures. We're also interested in their functional modularity and their mode of action to eventually be able to then make application ready, give them application ready properties through protein engineering, and potentially make hybrid toxins. and again, understand their mode of action. There's another engineering goal that I already mentioned earlier, which involves this like toxin identification. So I call it an engineering goal because, as I said, these sequences of these toxins seem to have either diversified or were never related to each other. So we also find like really good means to identify the toxin coding genes in yeast genomes, ideally later on in meta-genome samples, such that we don't need to really go out there and do environmental sampling anymore. So this is what we envision. And what I want to do now is really talk about a bit more in detail about two aspects of the pipeline that we are already well established. And this is the accessing toxins and then understanding them. And we were actually able to work on both of these aspects in parallel. The reason we wanted to work on them in parallel is that we already had some toxins sequences available to us, so you can just find them in databases. But most of the toxins that were available to us had really not favorable properties for applications. So they had a very low and strict pH requirement and temperature requirement. So what we wanted to do when we started to accessing new ones is really to map what is out there in terms of, potentially new toxin, and also if you find better templates for certain applications. So templates that, for example, have a better pH range. I also want to mention here that although I'm a bioengineer, and although when I started, I envisioned that we would already be able to do a lot of engineering, once we started this project, once I started this whole research program 5 years ago in Groningen, I realized that we understand so little about these toxins that we actually had to step back first and kind of make an effort to understand what's going on. And I think now at the end of the talk, you're going to see we're now ready to engineer these toxins. So it's great that I'm also moving now to a bioengineering department, because that's, of course, what I want to focus on in the coming years. Okay, so talking about accessing, so antifungal yeast and where to find them. So this is work that is on bioRxiv by now, and was done by a graduate student in my lab, Alicia. And what she did, she really went out there and she sampled yeast from over 1,000 samples. She isolated about 680 strains, also collected microscopy data, and then species identified them using a PacBio sequencing approach. And we have a really diverse collection of yeast now with over 150 species, which actually was just submitted to the Westerdijk Institute, which is a biodiversity institute in the Netherlands, such that those yeast are available to the community to be used. We kept back those that are interesting to us, because those that have antifungal properties. So we then screened them against 8 relevant fungal pathogens, those relevant for food spoilage, but also crop pathogen and several human fungal pathogens. And then just summarizing what we got out of there. So a high number of these yeasts, so 31% of these Dutch yeast showed actually antifungal activity against at least one tested pathogen. That means that now in our lab, we have over hundreds of isolates with antifungal activity against plant, food, and human pathogens. Interestingly, they had very different modes of action from broad, not modes of action, but let's say, target ranges. So from broad to narrow. So some of the yeast really were very specific. They only target one of our panel of 8 pathogens, and some were more broad, so they could kill all of the fungi. And we had everything like in between. So we have a really diverse application area now covered, I think, with these yeast. We also did some preliminary studies into the different classes of molecules that are likely secreted by these yeast. So a huge portion of these yeast actually secretes iron-chelating compounds. We call them siderophores, which we didn't expect, but we're now also interested in my lab to actually explore how they are made, like the biosynthetic pathways, and how we can further kind of study and functionalize them. About 30% are actually proteins, so those are the ones we're interested in. And then about 10%, a bit more than 10%, make likely small molecules. And then also in terms of templates, so we find when we tested the pH activity profiles of these protein compounds, we already found some interesting pH profiles. So proteins that work at higher pH than the templates we already had at hand, which is interesting for certain application that require a broader pH range. So also mentioning that we want to actually apply those toxins later on in the food chain as biocontrol agents or for food preservation. So we also did an exploration into actually food products, and Alicia isolated yeast also from just daily consumed products like supermarket food and beers and other yogurts, all these products where we know yeast are a part of it. So we have over 100 isolates from daily consumed products. And also here we actually find that we have a lot of products, especially wheat beer, where the yeast component in it already secretes an antifungal compound. So this already tells us that likely those could be safe for consumption. Also, in a similar way as previously shown, these yeasts show targeting specificity. Some of them having like very broad activity, and others being like very specific against certain pathogens. So that was interesting to see in terms of applications. So ongoing work is now to actually work on this workflow that I had mentioned earlier, where we try to identify how these toxins are actually encoded. So here we do use full genome sequencing and proteomics of the secretome to kind of match what is actually secreted, to identify potential open reading frames, then functionally verify them in yeast. And the final goal would be to eventually have a larger set of a toxin database, such that we can then use it for learning to directly find these toxin encoding genes in genomic data, rather than going out there and like really finding yeast. And this is ongoing. In the same way, what is ongoing is that we identify or try to understand the diversity of modes of actions encoded in these toxins. And here we use genome-wide CRISPR ice creams and growth selections to find gene products that are involved in a certain toxin's action. And that will also, if we sample that across all the different toxins, it will give us basically a foot in the door to understand how different these modes of actions are. As I mentioned earlier, it's really important in the field to have different modes of actions at hand, such that we can either learn from them and find new Achilles heels to kill fungi, or directly use the toxins, for example, in health applications. So next, I want to talk about this understand area. As I said earlier, the goal, or why we want to really understand on a molecular level how these toxins work, is because it will enable us to later on engineer them. The major player of this section of the talk is this toxin K2. So this is a protein of about 376 amino acid, which is processed into a mature toxin that is secreted, that is chopped into an alpha and a beta domain that come together. So this precursor is further processed. And this K2 gene is naturally encoded on a double-strand RNA microvirus in Saccharomyces cerevisiae, but we, of course, clone it on a plasmid to work with it because it's just like easier. And then there's something known about the mode of action from older literature. So it first actually docks to the fungal cell wall via beta-1,6 glucan. Then it likely docks to a second plasma membrane receptor. And then it disrupts the cell membrane just by punching a hole in it. We don't know if it makes a defined pore or if it just inserts in the membrane, but cells die through to ion leakage. And the target range is rather narrow, so it mostly targets actually Saccharomyces cerevisiae, but also other closely related species that are human pathogens or food spoilers. So as I mentioned earlier, we want to know how the antifungal activity is encoded in this toxin. We know a little bit, so we know a bit the mode of action, but we really don't understand which amino acids are responsible for this action, and if this action is actually maybe modularly encoded on this open reading frame. So we do know a bit about the precursor, or we can deduce some, cues from the precursor. So as, for example, processing sites. So, as I mentioned earlier, this protein is processed when it's passage through the Golgi, and we have, we can predict this processing sites. But we really don't know if we really understand these domain boundaries. So what is the domain boundaries are actually important to know how the mature toxin looks like. We don't know if the function of these toxins, so the docking and the cell wall disruption, are actually modular. We don't know its 3D structure. So that has been a challenge actually in the field to get enough of these toxins, as so little is secreted by these yeast often, to make structural, to do structural analysis. We also don't know how these two domains actually hold together. So that might be by disulfide bridges, for example. And we don't know how amenable this whole protein is for engineering. So what we do to approach these questions is, in my laboratory, we do sequence function mapping via mutational scanning. So that means we create a library, for example, either an alanine scan where we just replace each amino acid by an alanine, or we do deep mutational scans where we replace each amino acid by all the other amino acids, or we do deletion scans, so all types of libraries. And we then have functional screens in the laboratory. Here I illustrate one of them, which is a kind of a screen that screens for overall function, where we basically use liquid handlers or robotic system to pin each variant of the library onto a sensitive indicator strain. And then this indicator strain basically indicates sensitivity by making this halo assay. And then we have some image analysis tool that then tells us how big this halo is. And this we can use to then basically link phenotype and genotype. So we bin according to functionality, and then we just sequence to get basically a score for each amino acid in the protein sequence. And that gives us these kind of nice maps. And this is a sample map for the toxin K2. So this is still unreviewed data that I'm showing here very briefly. So one way to look at it is this kind of like simple sequence function map, where you can see which portion of the protein is actually mutational tolerant. So these sequences here have shown little loss of function. We also can see these like functional hotspots, so like areas where that are really important for the function. And we were already lucky because we could find some gain of function mutations in these simple alanine scan here, where we already would see higher activity. There's also other ways to plot this data, which gives you a more global view on how tolerant this precursor is to mutations. And just briefly walking you through what we can learn from these data, in addition to more experiments that we usually do being inspired by these data. So first, it helped us to refine domain boundaries based on essential and non-canonical processing sites. So we now understand better how this protein is actually processed. And this understanding was really important to refine structure predictions of the mature toxin. And this then helped us using AlphaFold, of course. And this then helped us to also better understand how these two domains are hold together, which is not by intradomain, which is not by disulfide bridges, but rather by salt bridges. And it also actually showed us that there are some structural similarities, apparently, with other toxins that are out there. So this, again, comes back to this evolutionary history. So there might be actually now a structural way to group these toxins together and potentially use that to identify them in genomes. Also, in terms of engineering of K2, as I said earlier, we're interested to making it, for example, more change its specificity in terms of target range, but also making it broader in pH applicability. So first of all, and also modularity. So first of all, this alanine scan, again, in addition to other experiments that we do, really showed us that this precursor is modular. So we now know this is alpha subunit is actually encoding the toxic function. We also have suspicion or good indication that the beta subunit is responsible for docking. And we also know that now that this signal peptide here, actually, so the N-terminus encodes for the signal peptide, but also this like self-protecting immunity. And I'm going to talk about this in a bit more detail in a second. So modularity can be extracted from this data. We also have some clues now about also why the protein is so sensitive to pH. And this is mostly derived from this other toxin that we can compare it to. And likely it's because of charge distribution. And that gives first cues and kind of how we could engineer it to make it better work at higher pH, because currently it's really switched off at a higher pH than 6. We could also already elicit some new functions just with like single mutations. So we have some some mutations or some residues that when changed to an alanine lead to really higher activity. And we also have indications that we can really toggle the functionality. So make it, for example, more specific towards Saccharomyces cerevisiae. So here you, for example, have the wild type toxin. If you just change one amino acid, it still stays active against Saccharomyces, but loses activity against another fungus. So this is first indication that we will be able to engineer these toxins, and also indicating that these alanine scans or deep mutational scans are actually a really good way to probe and understand these toxins. So next, And last, actually, I want to talk about this modularity and specifically this immunity function. And that's something that we just published and really work that has been done or led by Rianne. Rianne was also the graduate student that was doing all the alanine scan that I showed before. And what she was interested in is actually in this question, how these carrier yeast, so these yeast that make these antifungal proteins, can protect themselves against the toxins they secrete, which is also important for bioengineering, because later on, if you want to express them in heterologous system, you need to have a way to make producers that don't self-intoxicate. So just a small experiment that we can do here on the slide, so you understand what I'm talking about. So imagine that you have a yeast, your favorite laboratory yeast, for example, BY4741. You transform it with a plasmid that encodes for your toxin. So the cell then starts secreting the toxin, and as soon as it meets another cell, and that is also again your favorite yeast strain in the lab, that has not been transformed with this plasmid, this yeast strain will be sensitive. And you can see that here in this like very simple experiment where you dot a bit of this toxin producer on a sensitive indicator, and you see that halo here. On the other side, if you do the same experiment with a plasmid containing yeast strain or a toxin secreting yeast strain as the indicator, you see that this strain is actually immune, meaning that the gene product of K2 itself, so just making K2, the K2 open reading frame is necessary and sufficient for this immunity phenotype. So Rianne was really interested how that works, and also, first of all, where this resistance factor or this immunity factor is encoded, if that is modular or rather kind of like diffuse across the protein precursor. So what she did here, or what she did to approach this, is she did a small actually deletion scan, and that is then related to a similar workflow, but here we use a different assay. We actually use an immunity assay. So here she was just interested if she adds external toxins to these deletion library, would she then see a strain that is resistant, or would she see a strain that is sensitive? So you basically had this on-off behavior as a readout for our assay. We were prepared to create a larger library, but we started with a smaller library where we really just deleted big portions of the protein, 27 amino acids. And that already was kind of successful in guiding us into the right direction, because she made these 16 constructs where she just deleted, portions of the protein, and then she added external toxin and just measured which of the protein precursors would still encode for this immunity. And we were like very quickly guided to the N-terminus, because if we deleted the first one of the first segments here in the N-terminus, we basically lost complete immunity, while the other deletion constructs were still harboring all the immunity. She then turned around this experiment and basically just produced N-terminal fragments, so from 108, and then she kind of like trimmed that down to produce shorter and shorter versions of the N-terminus to kind of find this cutoff where we wouldn't see immunity anymore. And we could really define that you need 47 amino acid from the N-terminus to gain full immunity. So basically a 47 amino acid N-terminal peptide is sufficient for this immunity or resistance phenotype. She also did a bit more trimming to really find the minimal peptide required. So she trimmed on the N-terminus and the C-terminus, and then we could basically identify a sliding window of amino acids of 39 amino acids that would then fully encode this resistance or immunity factor. She also showed that this is actually concentration dependent. So you need a certain amount of this immunity peptide to be made in the cell to actually make it resistance. And if you make it too short, then actually it has less efficiency in being protective. So there's something stoichiometric going on with the amount of this immunity peptide being required. So now we knew where this immunity was encoded, so somewhere here in the N-terminus, 39 amino acids, but we still wondered how it actually works. So what is the mode of protection of this peptide? And the first thing that we saw is that actually, or what comes to mind when you think of the N-terminus of a secreted protein is, of course, that it likely also encodes the signal peptide. So the signal peptide is this peptide that leads a protein into the secretory pathway. And in yeast, this is usually not sequence conserved, but rather just like feature conserved. So usually it has this like hydrophobic H region here in the middle, and then a positively charged N terminus, and then a C region where the cleavage site is. And when we look into our immunity peptide here of 47 amino acid, it actually has exactly these features. So we basically thought that, the signal peptide also encodes for this immunity. So she tested if this is the case. So she basically took this sequence and, added it to a different protein. In this case, we used a different toxin to also show that if we just replace the original signal sequence of this other toxin by the one from our toxin, we would see secretion. So this indicates for us that this signal sequence has actual dual functionality, which is something that is rarely reported actually in literature. So it first targets the toxin into the secretory pathway, and then it actually ensures toxin resistance. So it stays back in the cell somewhere here in the secretory pathway or in the plasma membrane and makes sure the cell is protected from the toxin. So this still didn't tell us exactly how this works, because we now know that what distinguishes a resistant cell from a sensitive cell is that the resistant cell just has this peptide somewhere here stuck in its secretory pathway, but we still didn't know what of these steps it actually protects from. It could be docking to the cell wall, it could be docking to this other membrane receptor, or it could also be at the level of like punching a hole in the cell wall. And we did a lot of different types of experiments. I think we have it narrowed down now to knowing that this immunity peptide actually directly interferes or prevents the toxic this insertion into the membrane. We can show that by showing that if we just express this toxic alpha subunit here, and we co-express the immunity peptide, then we can basically protect from the toxic function. So we have good indication that the immunity function directly protects from this toxic function that is encoded in the alpha subunit. So this is now also bringing me back to basically the more synthetic biology applications we have in mind, because we now showed that this K2 is really modular. We also showed that we can really split it into two different open reading frames. So we can separately express this immunity function, and we can separately actually express a toxin that is secreted and needs co-expression of this immunity function. So really a split version. And we have different varieties of this antitoxin that available to us. So depending on the lengths I have said, it has different capacity to protect the cell. And Rianne was also able to actually engineer different versions of this toxin that are more toxic to the cell than others. And these we now kind of put together or have experimented in using them as kill switches for biocontainment or potentially also for product release from cells. And we're also using it as tools for building synthetic yeast communities. So this basically brings me to the end of the talk. So I hope I could show you a bit that we have a nice pipeline where we can access, understand, and hopefully soon engineer these toxins. As I mentioned earlier, in the last 4 years, we mainly focused on this understanding area, and now we're kind of ready to really hone in and see if we can engineer these toxins. And as Tae-Suk already mentioned, soon we're moving to Imperial College. There, we're also going to work on all the other aspects that are part of my research program. So if you're interested in it, watch our space for what's coming out here. And with this, I really want to thank my lab, all the people who made that possible, but also our funding agencies and all our collaborators, and you for listening to my seminar.
Fantastic, fantastic. I learned a lot about fungus and because I also not brave enough to enter that field. Fungi always kind of make me nervous because that contaminate every single culture. When I was a master student, that's almost 27 years ago. So since then, I scared about fungi.
What are you afraid of? That the fungi will infect you?
In fact, my bacteria culture at the time,
Okay.
basically one of my lab mate, dealing with the fungus, and then everywhere that basically contaminates. So since
Although,
then but I believe that there are two type of the fungus. I believe fungi. One is kind of, you know, everywhere. The other is kind of stay there. That's what I heard recently. So now I'm a little bit more comfortable.
Yeah. Although I have to say, as fungi usually grow much slower than bacteria, we have rather the opposite problem. So that
Oh,
that we get maybe bacterial contamination. Or once we did the isolation, for example, we needed to actively get rid of the bacteria or make sure that we actually enrich for yeast and not for bacteria.
I see. So, but that's in that case on my long belief because the at that time, many years ago, I terrified by that's clearly I can see by bare eye. That's the problem at that time, I believe. So, Chimeng, do you have any question, comment?
There's actually a question in the Q&A.
Yeah, actually, I have the question in the Q&A, but I want to give you the time first, and then I go to the question from Canyon.
I guess it's a bigger picture. So when you try to find drugs against fungi, how can you systematically test for toxicity, like in vivo or against like other cell type?
Yeah, that's a great question. So you actually have to test them against mammalian cells in like cell culture, for example. So one of the reasons we're also interested in these toxins, and actually it relates very much to Kenyon's, did I pronounce that name? Yeah, Kenyon's question, because he also asks, how can you actually target specific cell types? And why wouldn't you target human cells? So maybe I can just answer this question altogether. So, well, you never know. Like, of course, with any drug, you need to make sure you don't target human cells, and you can only know by testing it. So you have to test it in cell culture. But the reason we're also interested in these yeast toxin is because usually they have this docking function where they're forced dock to the fungal cell wall, and the fungal cell wall is, of course, specific to fungi. And we think they first need to accumulate there and maybe enrich and can then act on or like unfold their toxic function. So one reason I'm interested in these toxins is because I assume they will not have toxicity against humans because they have this docking function first. But this being said, we have to test it, and this is why in my lab for applications and for industry collaborations, we usually first go to biocontrol and food. Given many of these toxins are already part of food. So for example, first of all, I mentioned that in many food products, we also find these antifungal compounds from yeast. And second of all, for example, wine, the major commercial wine strains actually encode these toxins. They were purposefully breeded into them to make them kind of, already use them, let's say, as food preservatives, like as beverage preservatives. So we already have an indication that they're actually safe to consume. But further toxicology studies would be required to really prove that.
Do you have a favorite workflow or human cell type or kind of a assay to test for toxicity? Or
yeah, so
does it have to be some kind of co-culture system, or it has to be like mice or actually animals?
Yeah, so I think I'm not an expert in that. So as I said, we mostly focus on these biocontrol and food applications at the moment. So whenever I kind of like pitch it to funding agencies, I would rather think that we want to get inspired by all these different modes of actions to maybe then have small molecule drugs that can mimic these mode of actions. So, so me in the lab, I have no workflow for actually testing toxicity in the lab. So we're not a lab that does a lot of mouse studies. But I think how people do it is actually first just human cells, and then later on maybe even organoids, and later on like mice studies. Yeah.
Thank you.
Okay, so I wanted to ask some naive question because I just curious. So you mentioned a lot of toxin that coming from fungi or yeast. The question I have is, so this kind of toxin is basically harmful to even including bacteria?
Yeah, I think it comes down to the same question as if it's toxic to humans, is it toxic to bacteria? So some of them might. So for the ones that we have tested, we haven't seen any that is active against bacteria. But in literature, you find also yeast that can kill bacterial pathogens. But again, for the ones that we closer work with in the laboratory, where we study the that we really study on a molecular level, they have this docking function, so they need to dock to the fungal cell wall, so they're not active against bacteria.
So some toxin, toxic to bacteria, but some, toxin toxic to the eukaryotic system, eukaryotic cell, basically.
Yeah, I mean, you also need to think about it that these toxins are really broad in terms of everything, in terms of, their sequence, their function. So as I mentioned earlier, they don't seem to be evolutionary related to each other.
Yeah. So I'm asking
the question you're answering, I think for each toxin, and actually we stopped calling them toxins because it doesn't sound great in the beverage industry if you call something a toxin. We start calling them mycosins. So as in relation to, for example, bacteriocins, that's also a bit how you how you can think of them if you know the field of bacteriocins. But just to paying tribute to the historic names of killer toxins, and also to find that literature, we keep that name. But you need to look at each of them separately because they're not sequence or function conserved, and each of them might need to be evaluated independently to answer, are they active against bacteria, yeast, humans, so on. So for the ones we work with, we know what they're active against, but
got it.
yeah.
So but basically the producer should have also antitoxin, otherwise they will kill themselves. The reason I'm asking that question is I'm very interested in the kill switch development, but I now see huge potential. You may find many pairs toxin antitoxin. At the end of the day, I could use that one for the kill switch function,
Yeah.
or some other thing like plasmid without antibiotic resistant gene. That's another interest I have. In that, if I use the toxic antitoxin that's coming from the eukaryotic system, not from bacteria, they might be orthogonal.
Yeah, you're absolutely right. That's also the vision I had and the vision I still have. It's just when I started, I thought we can just do it, but then I realized, okay, we just don't fully understand how all of this works.
Okay.
But you're absolutely right. I think they might be great for any of these applications.
Absolutely. So
Biocontaminant plasmids.
let's collaborate. But
we can also talk, and I would be happy to discuss some ideas.
yeah, let's collaborate one day. I mean, someday. I'm very interested in this system now. So fabulous. Cheemeng or anyone have more question? Let me double check whether we have more question from the audience. We already answered, I believe, the question from Kenyon. Actually, by the way, the question was just to other audience. I mean, the for K2, how does it target specific cell type? If it is toxic, why wouldn't it target human cell? And then I believe the Sonia already answered that question. If we don't have any other thing, let's close, and then we just probably chat more if we wish. Okay, so thank you all for joining and staying today. We'll meet again on November 18, Monday. Just please note the different date at also different time, 11:30 a.m. Central Time, the same Zoom link. Note the time and date change due to the time conflict actually with one of the speaker. Actually, I met him today, Tobias Erb. We'll have the Professor Tobias Erb right now in Brno with me. Of course, but he's from the Max Planck Institute for Terrestrial Microbiology. Also, Professor Jaya Joshi at University of British Columbia, Vancouver, Canada. As usual, the follow-up informal chat will occur without recording. Please stay here if you are interested in chatting with us. I'll promote you to panelists who can speak and show your handsome and pretty faces if you wish. And thanks. I will stop recording. Just give me 1 second.